this video is going to be a bit of a mishmash of different ideas. We're going to start by talking about how animal viruses are cultured, then move on into the teratogenic effects of cancer viruses and the virus-like agents that are, they're similar to viruses, but not quite exactly the same. Oh, and by the way, teratogenic effects, those are developmental issues that, so as fetuses are developing, sometimes strange developmental issues arise, and viruses are known to cause these kinds of teratogenic effects. So we'll take a look at all these things now. So there were two discoveries that made it possible to culture animal viruses, and this led then to what is sometimes, like everything has to have a golden age in this book, which I find a little amusing. So this led to the golden age of virology. So these two discoveries are first that they figured out how to free individual cells from a tissue without damaging the cells. And it requires an enzyme named trypsin that can go in and break down the proteins that hold individual cells together, but they don't break down the proteins that are in the cell membranes. And so we can now grow isolated individual animal cells in culture, which then opens up the possibility of infecting them with viruses and seeing what happens. The second of the discoveries was that you can use antibiotics. So in the 40s, as penicillin was one of the first antibiotics that was made and developed that was actually effective and didn't cause lots of other diseases. Remember our sulfa drugs from the 30s. So in the 40s, we began developing antibiotics, and these antibiotics can be used then in these cultures to prevent bacteria from coming in to infect them, so that we can allowing the scientists to only see the effects of viruses on the cultures. So this led again to that golden age of virology from the 50s through the 60s, during which more than 400 viruses were isolated, described, and studied. And as with phages, we use these plaque assays then to identify cell lines, tissue, like animal cell lines, that are susceptible to particular viruses. And then we can study the effects in those cells based on viral infection. So what are some of the techniques? Well, first you make a primary cell culture. This is the first culture that's grown from the cells that are taken from the animal. So you take the cells, you isolate them from each other, and then start growing them in primary culture. And then over time you can subculture that, so take a small sample from the culture that you grow, the primary culture, and then continuing taking small samples, growing them out, taking small samples, growing them out, and over time you can create a strain of the culture that has only one cell type in it. And so you get this monoculture. We call it a monoculture because there's only one cell type in it, of genetically identical cells through subculturing. And one of the important ones that was developed were the diploid fibroblast strains, and they are now among the most widely used cell strains for viral studies. So using fibroblasts, recall in connective tissue, fibroblasts are the cells that make the protein fibers. And if you're lucky, every once in a while, you can discover a cell that will reproduce almost indefinitely. So for many cell cultures, there is for many of these strains, there's a top number of generations of cells you can get from them. But there are some strains that seem to have no top number of generations. They just keep reproducing over and over and over again. And these are described as continuous cell lines. And probably the most famous of these continual cell lines are the HeLa cells. They were taken from a cervical cancer not biopsy, but when Henrietta Lacks, an African-American woman in the 1950s, came down with cervical cancer. They did a hysterectomy to her and then started culturing the cells from the cervix, by the way, without Henrietta Lacks's permission. which made for a very, very interesting story. It's called The Immortal Life of Henrietta Lacks. I encourage you to consider reading it because it's a really fascinating look at first the uses that the HeLa cells were put to, and then at some of the history of how medical research is done, and how some of the things that we now consider to be unethical that were done back in the day. Anyway, so the HeLa cells, named for her, first letters of her first name, first letters of her last name. These HeLa cells have been, I remember hearing about HeLa cells back when I was in college in the 80s. People talking about, how they were used to do a lot. I mean, so many different kinds of research have been done with those cells cultured from Henrietta Lacks. So in culture, when virus cells get infected, they frequently exhibit some very obvious, diagnostic even for some viruses, changes to the cells, referred to as the cytopathic effect, or CPE. So sometimes the cells will accumulate fluids and they become bubbly, like big and round. So you can see here some of the swollen cells, normal in this picture, swollen in the other, where they have accumulated those fluids. Frequently the cells will lyse and just die. Sometimes the cells will fuse together, forming giant cells that are multinucleate, and they're referred to as syncytia. So that's a weird term, but maybe you'll remember that from anatomy and physiology, where we talked about the atrial and the ventricular syncytium, all the cells that contract together. Here we mean syncytium from the standpoint of one gigantic cell formed from the fusion of a bunch of individual cells, like we see here. And then the last of these cytopathic effects is transformation, where sometimes the cells will convert from being normal cells into malignant or cancerous cells. And that's the subject of the last PowerPoint presentation. So I encourage you to make sure you get a good look at that too. This idea of transformation and cells converting to being pathogenic and malignant. So some of the cells, or some of the viruses that we see in infecting humans and other animals, exhibit teratogenic effects. These are induced defects in human embryos because of viral infection of the cells. Or we also see, of course, teratogenic effects in other animals, but medical science is most concerned about the teratogenic effects in humans. And this happens because some viruses are capable of crossing the placenta and infecting the fetus. And it turns out the earlier in pregnancy that this crossing, that this infection of the fetus occurs, then the greater the teratogenic effects, if it has one, because of all of the cells that become infected with these viruses. And as we've seen, there are some viruses that are capable of becoming latent in animal cells, and then any offspring cell from that cell is going to carry the virus in it. Some of the characteristic or some of the teratogenic viruses include cytomegalovirus, which is a very large virus, and it can cause mental retardation and organ damage. The herpes simplex viruses, which will cause damage to the eyes and to the central nervous system. Generally, these herpes viruses are given to the fetus very late in its development, as it is being born, as it passes through the vaginal canal, but still significant damage can be caused to the central nervous system and to the eyes because they are still in the process of developing. Rubella, also known as German measles, can cause deafness, sensory organ, and other damages to sensory organs, again, because of the development that still is ongoing, even after the child is born. Heart and circulatory defects may develop, and mental retardation. And then last, we've heard a lot in the news this year about the Zika virus, which is a mosquito-borne virus, causing microcephaly in infants, as, after when they're born with microcephaly, which is a very, very small brain and a characteristically small head. So here is a child with microcephaly. And so that leads to severe mental retardation and associated effects. So these are children who will likely never be able to care for themselves in any real significant way, and very difficult childhoods and adulthoods for these people. Okay, so those are the teratogenic effects, or some of the teratogenic effects we see associated with virus infection. And then this section will wrap up talking about some of the non-virus but virus-like agents that can cause disease. So the first one of these is a satellite. A satellite is just a small bit of single-stranded RNA that, when it infects a cell, has to have a helper virus assisting it. So it cannot reproduce. It doesn't have all the enzymes required to reproduce the same way that a full virus does. And so it occurs both, it must occur in an infection along with one of these helper viruses. And they are only found in plants so far, although there's speculation that they are in animals as well. So we see these satellite diseases in plants. And here you can see a plant, one that is protected from a satellite virus and the other that was diseased. The hepatitis delta virus is similar to a satellite or a viroid. We'll talk about viroids next, but it is the smallest of any of the virus or virus-like molecules, just a tiny scrap of single-stranded RNA. And like the satellites, it requires a co-infection with, this time, the hepatitis B. Let me try that again. Hepatitis B virus in order to replicate. So because it's so small, I think that's one of the reasons that the book is reluctant, or the authors are reluctant to call it a satellite. But I think you could probably suggest that this might be a satellite as well. So it has to have this helper virus, hepatitis B, in order to replicate. And what we see is that the increase in death rate goes up 10 times in people who have a co-infection of hepatitis B and this hepatitis delta virus over when you compare it to people who are just infected with hepatitis B. So it really significantly increases the lethality of hepatitis B virus, which is, of course, a virus that affects liver cells. So virophages are another of these virus-like agents. These are satellites that, rather than causing damage to the cell that they infect, actually reduces the pathogenicity of its associated helper virus. So virophages, like we talked about bacteriophages, well, this is not an infectious agent that will kill the helper virus. It reduces the pathogenicity of the helper virus. And they've been discovered, at first we thought there were just a few of these, and then we've discovered that they are everywhere. So we see these virophages that reduce the ability of the virus to affect the host cells. Viroids are small particles of RNA. They differ from viruses because they are not, they're very, very small, so between 246 and 400 nucleotides long. They're found inside cells, usually in the nucleus, but they don't have a capsid or an envelope around them. They don't produce proteins, so it's unclear exactly how they cause diseases. They don't require a helper virus. The viroid RNA is only copied in the nucleus, and they're hard to find. And so they've only been discovered in plants so far. They're known to be transmitted by seeds. So in the seed there will be this viroid there. And we know that aphids are also capable of transmitting them. As they suck the juices from one plant and then go and suck juices from another plant, they can carry that viroid with them. And it's believed that they function by interfering with the production of messenger RNA. And if that happens, then they either prevent or reduce the ability of the cell to produce proteins. So viroids, they're kind of just this bizarre little piece of RNA that may be interfering with messenger RNA production. Prions are especially cool. Prions, they are named from the fact that they are proteinaceous and that they are infectious. So these are normal proteins that are found in all, in cells, often in the nervous system, also in lymphoid tissues, but they will refold. So if you recall from a biology class, proteins have this very elaborate structure: primary, secondary, quaternary, and tertiary, and then quaternary structure. So prions refold, they change their secondary and tertiary structure, producing a non-functional protein. And what's interesting about them is that you get them, or they can spread within an organism, because once one protein refolds in this dysfunctional way, other proteins around it become more likely to also unfold and refold inappropriately. And so you can pass these prion diseases by consuming the brain, especially, of other animals that are already affected by the prion disease. And so in mammals, this is where you find it, is in mammals. So that's us, humans and monkeys and dogs and cats and rats and mice and cattle especially, and elk and other grazing animals. These are all mammals. When these proteins unfold in a strange way, then they create dysfunctional proteins, and that leads to cells dying, and you get this spongy characteristic to central nervous system tissue and to lymphoid tissues. And they can even cause cancers. We've discovered that if there's a protein called p53, it acts as a tumor suppressor in the cell. And if it unfolds and refolds and becomes non-functional, then that can cause the cell, because it no longer has this tumor suppressor protein acting, then it can cause the cell to become cancerous. And interestingly, prions are also found in yeasts, of all things. So what are some characteristics of prions? First, I told you about this strange folding or unfolding. So here we have the protein kind of forming this helical area, and instead of being helical, the protein unfolds and forms this pleated sheet kind of characteristic. And this protein unfolds and forms a pleated sheet. So in the change of shape, we change the function of the protein, which then will result in cells dying. So what are some characteristics of these prions? They're resistant to being inactivated by heat. So the protein is already denatured, essentially. So you can't really change it once it's denatured. They're not affected by radiation. Nucleases don't affect them because they're proteins. An enzyme that breaks apart nucleic acids isn't going to affect them. They are sensitive to other protein denaturing agents like phenol and urea, though. So heating doesn't mess with them, but if we try chemicals that denature, then we can see some effect. And they form, again, as these lengths of amino acids pair with each other. And so these links kind of pair against each other rather than being helical like they're supposed to be. And this creates protein fibers in the cell, and those fibers and the non-functionality of the cell, because it doesn't have these proteins, will eventually kill the cell. So what are some diseases associated with prions? They include Creutzfeldt-Jakob disease, named for two dudes who found this disease. And so Creutzfeldt-Jakob is not a cannibal disease like we see Kuru is. Kuru is a disease, or is a prion disease that was first discovered among native tribes, tribal peoples that engaged in cannibalism, that ate, say, the brain of frequently it was a person of high rank in the village who, as they died, so it wasn't about going out and fighting and killing and cannibalizing. Instead, it was a way of honoring the dead to eat their parts. And if they ate brain tissue of someone who had died of a prion-related disease, then that can stimulate prion-related diseases in the person who ate this material. Creutzfeldt-Jakob disease is genetic, so it's a genetic predisposition to prion activity. You've heard of mad cow disease, probably. That's called bovine spongiform encephalopathy. Bovine meaning cow, spongiform meaning, when it kills off the brain tissue, the brain gets kind of spongy looking. Encephalopathy. Sorry about that. Encephalo means talking about the brain, and pathy, of course, is pathogenic. Also, chronic wasting disease and scrapie in sheep. These are all prion-related, very similar diseases to each other. And it's not just sheep and elk and deer moose and cows that can experience prion diseases like this. Lots of mammals, and the book has a picture showing all that. And it can spread across species and to humans if we handle or if we eat infected, the tissues of infected meat and animals. And it can also spread through milk. So like the chronic wasting disease can spread to humans just because you're dressing the elk or the deer moose. And exposure to the organ meat can cause that to happen. Or handlers in slaughterhouses might also become infected with it.